In this lecture, we're going to continue going over the lymphatic system and body defenses. So we'll start by going over the humoral or antibody-mediated response. So B lymphocytes with specific receptors can bind to specific antigens because they recognize them. The binding event activates the lymphocyte that goes undergoes something called clonal selection. And a large number of clones are produced, and that's called the primary humoral response. Most of the B cells that are exposed to an antigen become plasma cells, and they produce antibodies that destroy those antigens. And this activity lasts for 4 to 5 days. But some of the B cells become long-lived memory cells. So those cells can live and circulate in your body for a long time, and so you can have immunity for many years. So in that secondary humoral response, those memory cells are very long-lived, and then a second exposure to an antigen causes a rapid response. That secondary response is stronger and it lasts longer. So it means that you can have immunity to something and be exposed to a pathogen for a second time, and the second time you're exposed, you won't get sick. So when a B cell is exposed to an antigen, the antigens are these little red dots in this case, and they bind to antigen binding receptors on the B cells. So you can see those receptors here. The B cell is then going to proliferate and it's going to clone itself, so it's going to divide. Most of the cells that it makes are going to be these plasma cells, and those plasma cells are going to secrete a whole bunch of antibody that can be used to deactivate more antigen. Some of the other cells are going to become memory cells. Memory cells can also detect antigen, but they can last for a long time because memory cells are long-lived. And so because they live for a long time, they're present in your body to detect antigen a long time from your first exposure. And so anytime they're challenged by the same antigen, you have a more rapid response. And so those memory cells are going to then divide if they need to, if you're exposed again to more memory cells, and then also plasma cells that will have, again, that rapid and strong response. So when people are vaccinated or exposed to an antigen, when you're first exposed to an antigen, you're going to have a response, and you're going to have an immune response over several weeks. So after you've been exposed, you'll have a response. Your peak response is about 2 weeks out from your initial exposure, and then it starts to decrease. But you'll never go back to zero if you've got those memory cells circulating. And so that way, with those memory cells circulating, if you're exposed again, and this is why we booster vaccines, it will increase the secondary response dramatically, so you'll be able to knock down that antigen or that infection much more quickly. There's two different things we talk about in immunology called active and passive immunity, so we'll talk about those next. Active immunity occurs when B cells encounter antigens and produce antibodies. Active immunity can be naturally acquired during bacterial infections, so that means you got sick with something and then your body has become immune to it because you were sick with it, and that can happen with bacteria and viruses. You can also have it artificially acquired from vaccines. The advantage to a vaccine is that you can become immune to a disease without having to have it, which is really important for certain diseases, especially diseases that have high fatality rates or have really injurious consequences. So that's why humanity has worked really hard to knock out things like polio, for example, because it can be very, very damaging to the people who have it. And so we vaccinated pretty much the entire globe and have almost eradicated polio. Another example would be smallpox, where vaccines have worked incredibly well to eliminate a disease. We have vaccines for lots of other things too, and anytime you have a vaccine, it's so that you can develop this active immunity. Passive immunity occurs when antibodies are obtained from someone else. So your body has not manufactured those antibodies, but you can have antibodies from another person borrowed, essentially, in a couple ways. So one of the ways that passive immunity is transferred is from a mother to a fetus. So when women are pregnant, some of those antibodies can pass from the mother to the fetus and give the fetus some immunity. You can also artificially pass immunity through immune serum or gamma globulin. And so this is when people who are immune to something can donate blood plasma and then they're able to use that plasma in people who don't have immunity to help knock down their infection. When this happens, immunological memory does not occur, so those B cell memory cells are not formed, but the protection is provided by those borrowed antibodies. So with another transfusion, for example, you would have more antibody and be able to fight the infection a little bit longer. Monoclonal antibodies are a tool that we use in science. So they're antibodies that were prepared for clinical testing or diagnostic services. And they're produced from a descendants of a single cell line. So they found a cell line that has a strong binding capacity for a specific antigen, and then they can use those cells to produce many, many antibodies. And they're used to diagnose things. So you can use them to diagnose pregnancy, for example, or they can be used for treatment after exposure to diseases like hepatitis and rabies. So acquired immunity can come from two sources. So naturally acquired immunity can be active immunity because you had an infection, or passive immunity from your mother when she was pregnant with you, or through her breast milk. Artificial immunity can be active because you've had a vaccine, or passive because you've been given an injection of some immune serum from another person. So antibodies are those proteins that are produced by the B cells. They're called immunoglobulins, or Igs, or antibodies. All of those are the same thing. And they are carried in your blood plasma, and they're capable of binding specifically to an antigen. So not every antibody will bind every single antigen. This is a molecular model of an antibody, and you can see it's kind of a Y-shaped molecule. So that's why they're drawn that way when they're not drawn at the molecular level. Antibodies are built of 4 amino acid chains, and they're linked by disulfide bonds. And so 2 of the identical amino acid chains are linked to form what we call the heavy chain, and the other 2 identical chains are called the light chains. And their specific antigen binding sites are present on each antibody. So in this diagram, you can see what a antibody looks like. So the heavy chains are represented in purple, and then light chains are kind of that bluish color, and they're held together by disulfide bonds. And then the antigen binding sites are at the top, where the 2 chains are parallel to each other. There's several different types of antibodies, so they all have slightly different jobs. So there's IgMs, and they can fix a complement. IgAs are found mainly in mucus. IgDs are important in the activation of B cells. IgGs can cross the placental barrier and fix complement, so those can pass from mothers to offspring. And then IgEs are involved in allergies. And there are several different ways that antibodies can neutralize an antigen. So they can do complement fixation, neutralization, agglutination, and precipitation. So this diagram kind of goes through some of those major ways that antigens are inactivated by an antibody. So antibodies can neutralize something by binding to the most harmful sites on a virus or bacteria. They can also agglutinate things, which means that an antibody can bind to several different things and kind of cause them to form clumps. They can precipitate things. So if something is soluble, they can become unsoluble when they're bound to an antibody so that it falls out of solution or precipitates. And they can also act as a complement, where they can change the surface of a cell in a bacteria cell, for example, by building like a channel protein, for example, that causes the cell to lyse. When the cell-mediated immune response happens, antigens have to be presented by macrophages to immunocompetent T cells. So that's antigen presentation. Sometimes those are called antigen-presenting cells because they show it to immunocompetent T cells. And then the T cells have to recognize self versus non-self. So they are able to determine which things belong to your body and which things don't. And then after antigen binding, clones form, as with B cells, but different classes of cells are produced. In this diagram, you can kind of see how that process works, because we've got the antigen presenting cell here, and then it's going to show antigen to the helper T cell, and then the T cell can activate cytotoxic T cells and communicate with the B cells. T cells can have a couple different jobs. So they're cytotoxic or killer T cells, and they specialize in killing infected cells. And they insert a chemical called perforin that causes infected cells to lyse or to break apart. And then helper T cells recruit other cells to fight the invaders, and they interact directly with B cells. So next we'll go through the function of those cytotoxic T cells. So first, the cytotoxic T cell will bind tightly to the target cell. So you can see the target cell is this yellow cell here, and the cytotoxic T cell is going to bind to it. Then the cytotoxic T cell releases something called perforin and granzyme molecules from its granules by exocytosis. So it dumps the contents that it needs to in between the interstitial space between the target cell and the cytotoxic T cell. And you can see here that those proteins are starting to be assembled. Those proteins are assembled to become these perforin molecules, and so they are able to form pores in the membrane of the target cell. Then the granzymes can enter the target cell through the pores that were manufactured, and it can start to degrade the cellular content. And finally, the cytotoxic T cell can actually just detach and search for another target because it does not degrade the T cell at all. So they are reusable, and every time they destroy a cell, they can go on to destroy another one. Then there are also regulatory T cells. They release chemicals that suppress the activity of T and B cells, and this stops the immune response to prevent uncontrolled activity. Because, of course, uncontrolled immune activity is not a good thing either. Sometimes we call it a cytokine storm, and it can also cause some pretty serious problems. And then a few members of each clone are also going to be memory cells for those T cells.